Hello, everyone, and welcome to phyloseminar.org. The current theme is single-cell analysis, and this is the first talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Dr. Reza Kalhor. Reza did his PhD with Lin Chin and Frank Alber at the University of Southern California, working on the 3D organization of the genome. He did his postdoctoral training with George Church at Harvard Medical School, working on technologies to store digital and biological information in DNA. Broadly speaking, Reza is interested in using molecular recording and genomic barcoding technologies to better understand dynamical biological systems such as embryos. Reza is currently an assistant professor of biomedical engineering at Johns Hopkins University. Welcome, Reza, and thank you for participating.
Thank you for having me, Eric. I'm very excited to be here. Welcome, everybody, to what is the first talk in a series of 3. I have been asked to, and I apologize in advance for the state of my vocal cords today, but I've been asked to basically introduce the problem set that cellular phylogenetics is concerned with. And I assume most of the audience is coming from a classic evolutionary phylogenetic background where the objective, as shown in this picture, is to understand the evolutionary relationship between species by comparing them. As we all know, all life on Earth basically originated likely from a single common ancestor hundreds of millions of years ago, which then evolved into all these species we have today. And by comparing their DNA sequences, we can decipher their relationship. Cellular phylogenetics is an analogous problem in that it tries to understand the relationship between various cell types that exist in a body. In the most exemplary format of cellular phylogenetics, those cells are cells in an animal that came from a single cell, the zygote at the time of conception, which then develops over, depending on the species, hours or hundreds or thousands of hours to form all of these cell types. Now, in the last 6 years or so, cellular phylogenetics has actually converged with evolutionary phylogenetics a lot, because we now have the ability to engineer the zygote to gradually accumulate mutations during development. And here I'm showing a zygote and these loci, each with a gray bar, which we can basically induce to, over time, accumulate these random mutations shown by colored pegs. And what that gives you in the end is a barcoded animal in which each cell has a set of mutations that speak to its phylogeny, and those mutations can be sequenced to infer a phylogenetic tree. And you all almost certainly recognize the similarity between this and evolutionary phylogenetics. And in fact, we don't even have to engineer the zygote, because at every cell division, there are natural somatic mutations that accumulate that are equivalent to these induced ones. And with the incredible sequencing power we have in this day and age, we can also sequence those and get the cellular phylogenetics without any engineering, though at a very high price tag. So in general, these approaches are referred to as lineage barcoding. Now, on the left, you see an example of a lineage barcoding outcome. Let's say a number of cells have been sequenced. Each row corresponds to a cell, and each column corresponds to one of those mutation sites. And the colors are the different mutant alleles. From that, you may naturally surmise that we basically need to infer a phylogenetic tree from this. And this becomes one of the important problem sets in this field, how to infer phylogenetic trees from barcodes. While it has a lot of similarities to evolutionary phylogenetics, it also has distinctions. These tend to be a complex set of mutations because some of these barcoding sites are very complex and work in interesting ways, and the patterns of mutations they create is not necessarily like the classic point mutation or that you might be used to. But you might also ask, why should we infer phylogenetic trees? How is that useful? And the answer to that question is that cell phylogeny is useful because of the ways animals develop. Here you're seeing the development of a human from a single cell, the zygote, that over time gives rise to this incredibly diverse array of cell types, which is in the tens or hundreds of thousands. So the zygote has a lot of potency, capable of producing all of these cell types. And in order to do that in an organized and reproducible fashion, it has to unfurl this potency, this potential, in a gradual, systematic way. And the result of this is that as the embryo develops, the diversity of cell types increases while they simultaneously become more specialized. And every time a cell becomes more specialized, it passes that information about its newfound identity or specialization down to its daughters through epigenetic mechanisms. And these epigenetic inheritance of cell identity is exactly why phylogeny is useful, because it speaks to how one cell's fate is determined and passed on to its daughters. So a great example of the usefulness of cellular phylogeny is in this very interesting model organism called C. elegans or the roundworm. C. elegans has 959 cells in every adult. And as you can see in this tree that I'm showing you on the left, every single C. elegans worm develops exactly the same way according to this tree. And every single division leads to two cells that have different fates. So in C. elegans, cell phylogeny and cell fate are one and the same. And this is incredibly powerful because, for instance, if you had a gene you wanted to study the function of, you could knock it out and see exactly which one of these divisions is altered. Or as labeled here, if you wanted to know what establishes neuronal fate, here are two sensory neurons labeled here and colored on the phylogenetic tree, you could go isolate these cell types at the base of this and sequence them and characterize them to know, to understand what establishes neuronal fate. But there's a huge difference between C. elegans and almost any other multicellular organism. For one, C. elegans has thousands of cells. Other organisms, mice and humans, for instance, tend to have billions or trillions of cells. C. elegans has invariant lineage. Multicellular organisms have a lot of stochasticity in their lineage. In C. elegans, all divisions are asymmetric, as I mentioned earlier. In most other organisms, there are more self-renewing, basically symmetric divisions than there are asymmetric divisions. So as an example of this, there's this very interesting recent paper from Kurens et al., where they did phylogenetic characterization using somatic mutations in humans. And this is the lineage tree they got from one human. And these pie charts here show the contribution of that cell in the phylogenetic tree to the entire human. And as you can see, the first two divisions after the zygote, one contributed 2/3 and another one contributed 1/3 to this human's body. What was interesting was that this was different, vastly different between different humans. And then if you look here with these arrows, I have labeled the testes seminiferous tubular cells, which have identical fate and identical history of ancestors, yet they have the maximum possible phylogenetic distance by being descendants of these two different cells at the beginning. The reason for this difference with C. elegans is that higher organisms develop through intermediate progenitor states. For instance, this is the early stages of mammalian development, where the zygote forms these potent mass of cells called morula. This is one progenitor state, which then becomes two different progenitor states, inner cell mass, ICM, and trophectoderm, and each of those then becomes later progenitor states. But the cells within one progenitor state are basically interchangeable, and in one organism, in each organism, a different set lead to the downstream progenitor states. In other words, in most organisms, it is this hierarchy of progenitor states that dictates development. And dividing cell and also captures biological insight. And dividing cells move through this hierarchy in a somewhat stochastic fashion. As such, phylogeny itself tends to be stochastic, tends to be sparse because we can't sequence all cells, and information poor because many divisions don't have cell fate information associated with them. So the second class of very interesting problems in this field is that how do we use cell phylogeny to learn something about these progenitor states? And this is the first talk in a series of 3 that talk about these 2 challenges that I introduced. The first talk today here, I'm going to talk about using time in phylogeny to reconstruct a hierarchy and dynamics of complex progenitor fields during development. This is work mostly led, almost entirely led by Wei-Xiong Fang, a phenomenal postdoc in my lab, who's also co-mentored by Hongkai Ji. But in the next few weeks, there will be 2 other very exciting talks, one by Sebastian Prio, who will talk about time-resolved trees to study the evolutionary dynamics of cell populations, and as I understand, how it relates to cancer, as well as Sophie Seidel in Tanya Stadler's lab, who is using phylogenetic approaches to infer cell division and apoptosis rates based on timescale phylogenies. One common theme that is emerging in the field, and I have highlighted here, is the concept of using time to make inferences about ancestral or progenitor states that all of these talks will talk about, but they are applying it in a slightly different context and different progenitor states. So for our section, my lab basically develops barcoding approaches. And this work was motivated by us asking ourselves, given the divergence between phylogeny and fate in mammals that I told you earlier about, and the stochasticity, and the fact that we can only sequence a very small fraction of cells, how useful barcoding approaches can be, and how do we, what types of barcoding would be more informative and useful. So to answer that question, the first thing we had to do is basically create a simple model of development, which we call quantitative fate map, which captures this hierarchy of progenitor states as labeled here by P, followed by numbers, that lead to the set of cell types we have observed in an animal or in an embryo, as labeled by T, followed by a number. And not only the hierarchy of these progenitor states, known in the field as their potency, is something that is very important, but also their individual behaviors are very important. For instance, in these quantitative fate maps, we can assign cell division rates and cell death rates to every progenitor state, and basically know at the time of commitment, here signified by these dots, how many cells existed, and then we can assign what fraction of them commit to each of the downstream states. This is called the commitment bias. We further establish commitment time, which is the exact time these cells commit to the downstream fates, as well as population size, which is the number of cells that a progenitor state had at its time of commitment.
Reza, can I just interrupt you really quick? I mean, just because I can see this is going to be an important concept. Do you mind going back to that crazy slide where you had the sperm cells coming from 2 different lineages? I just want to totally make sure I understand what's going on there, because I can see this is going to become important. So these are these little pie charts are these are cell populations and fractions.
This is individual cell. This is one cell. This is the other. The pie chart shows what fraction of the cells in the eventual human came from this, and what fraction came from that one.
So we should think of these 2 cells with the 2 arrows as being like 2 independent, sort of not very differentiated cells. Maybe they're a little bit.
No, they are not differentiated at all. They are identical in their molecular properties, yet they end up behaving very differently due to stochasticity.
Okay, so in the description that you were just sort of giving, they're not committed at all?
No, this is pre-commitment.
Okay.
These 2 cells are pre-commitment. Note that because so few cells have been sequenced, which is the limits of technology, we can really only see the first few divisions very well. So in the model that I am showing you now, we are taking this a lot further. So that tree is mostly in this stage and very sparse afterwards. In this model, we are going down all the way to a set of imaginary terminal cell types.
Okay.
And then we also assume either we are sampling them with a fixed strategy, meaning that, let's say, if you have neurons and muscle cells, you decide to take 100 cells from each, or you're doing a single cell sequencing type of approach, and you're sequencing as many cells according to their abundance.
Perfect. Thank you.
Right? And then we actually created a panel of these quantitative fate maps, 331, in 3 different size categories of 16, 32, and 64 terminal cell types. These are their respective numbers shown here. And within each category, from a low level of imbalance to a very high level of topological imbalance. And basically, each of these resembles a developmental scenario that we have seen. For instance, this is very familiar to people who work in eye development. Right? Now, the next challenge becomes, given a quantitative fate map and knowing the number of cells at each stage, how do we simulate phylogenies? And I'm not going to go into much details here because it is published. But the short answer is that we use a coalescent model in which, since we know the number of cells at each stage going backwards, at each stage, we estimate the number of their progenitors going backwards in time, all the way back to the zygote. And then we randomly connect each cell to its ancestor, resulting in a phylogeny such as this one. And every time we repeat this process, even based on the same quantitative fate map, we are going to get a different phylogeny, which captures the stochasticity that we will observe in real biology as well as barcoding experiments. So in all, we had these 331 panels. We instituted both the fixed and proportional sampling modes and repeated phylogenetic simulations 5 times for each, leading to 3,310 phylogenies, where 100 cells were sampled on average from each of the terminal cell types. And the results I'll show you going forward is basically things that were tested and assessed on this panel. Now that we have these models of development, the first question is, how do we recapture, having only this phylogeny, the topology of the fate map, which now at this point is not known? So for that, we focused on internal nodes in the phylogenetic tree, which indicate separation of fates, as we call them, fate separation or phases. And you see a subtree here. Each internal node is labeled with the eventual fate of its terminal cells. T4 and T1 means that this one gave rise to 1 and 4. Now, we call those internal nodes that indicate separation between 2 fates as phases. For instance, this one labeled here in orange shows a separation between fate 4 and 5, because 5 is only on one side and 4 is only on the other. It also shows a separation between 1 and 5, but I have only labeled here the nodes that indicate separations between 4 and 5. Now, when you look across the entire phylogenetic tree, these are those orange nodes showing T4, T5 phases. I have shown phases for another pair of terminal cell types. And basically, we can define a distance based on average, where these are seen relative to the leaves of the tree. And we call this the phase distance between 2 terminal cell types. We can define a matrix of pairwise phase distances between all pairs of terminal cell types, as you can see here, and then apply a hierarchical clustering, which gives us a tree topology, which is the infer topology for what would have been the quantitative fate maps topology. These are now inferred states. If the topology is perfectly resolved, they match the ones that were used in simulation. But the phases are basically using the specific patterns of fate restriction in the tree to define the topology of the fate map. And now this is what we call the phase algorithm. We tested its performance across the panel of 3,000 simulated experiments that I showed you, and you see the results are separated by fate map size as well as by sampling strategy, whether it was fixed or proportional. And on the x-axis, you see the imbalance of the tree, and the red line corresponds to the phase algorithm. The blue line corresponds to shared progenitor score, or SPS, basically one of the very few other algorithms that existed that doesn't use time, rather uses the observed potency in the internal nodes of the phylogenetic tree. And as a control for background, there are random topologies. And the measure of reconstruction accuracy is the Kendall-Collian distance, which is 0 between two rooted trees if their topologies are identical. And what you see here is that the phase algorithm actually performs very well for well-balanced trees, and even in it is still informative in highly imbalanced and very large trees. Now that we have the topology using the phase algorithm, the next step is to decipher important parameters about each progenitor state's behavior. One of them is commitment time. You might say that topology kind of gives you the relative commitment time, but in development, it's important to know, for instance, whether this progenitor state committed before this one or that one, because they can induce one another. The other is population size. How big each progenitor state is very important, as well as commitment bias. When this one commits or differentiate, what fraction goes to T1 and what fraction goes to T2. So to extract those types of parameters, the first step we took was assigning inferred states to all internal nodes. Now, having this phylogeny that the phase this topology that the phase algorithm gave us, we can annotate internal nodes of the phylogenetic tree with more information. For instance, if a node such as this one has given rise to T5 and T3 fates, when we look here, we see the only progenitor state capable of that is inferred progenitor state 8, and we can assign it its observed state. And we can do this for all of the nodes in the phylogenetic tree in greedy fashion. So the result looks something like this. This is the entire phylogenetic tree, and the inferred state of all internal nodes have been colored according to this key. When we look at these inferred states, now we get a better sense of the divisions that represent self-fate, commitment, and differentiation. And those are divisions in which a cell of a particular progenitor states give rise to two less potent cells. We call these inferred commitment events. They are labeled with asterisks on this small phylogeny, which you see here, for instance, 2 giving rise to 14 and 2, which are both less potent. So this is an ICE, whereas this one isn't, because 2 is giving rise to 2 other state number 2 cells. When you look across the entire phylogeny, for 2 different IP5 and IP8, I have colored all their inferred nodes, but the darker color are the ICEs, the inferred commitment events. And the timing of them in the timescale phylogeny actually represents the commitment time of the corresponding progenitor states. And if it is committing earlier, its ICE times are taking place earlier.
Sorry, Reza, can you go back to the previous slide? I didn't quite follow that. So I understand. So I think that the ICEs are like IP when you get from IP2 to IP14.
Yes. So
but so how do we? What does it mean to have? How do we have signal to understand 2 lineage commitments that are sort of the same type?
So based on the topology we have inferred from the phase algorithm, right? We know IP2 is here. So when I look at this cell, I look at all the terminal. These are observed cells. I know this is, for instance, a neuron, and that's a muscle. So I can say the only state this internal node can have is IP2, because that's the only one that can give rise to both 2 and 3 and 4, as you see here in the topology, right?
So if you don't mind if I recap. So we take all the cells, we build a tree, then we use that to build the reconstructed fate map topology. Then we take that fate map topology and redecorate the original tree with the fate maps.
Exactly. This is an iterative process. First, we get the topology with phases, and then we use that topological information to further inform the phylogenetic tree, right? And then we get the ICEs out of them, which are a better representation, more comprehensive. They tend to be more abundant and more informative than phases regarding commitment times.
Perfect. Thanks.
Thank you. That's a great question. So basically, these ICE times the average of the ICE times we see across the phylogenetic tree for a progenitor state represents its commitment time. And when we look at our panel of 3,000, this is what the performance looks like. On the left, you're seeing fixed sampling. On the right, proportional sampling. X-axis is the commitment time that was simulated, the true time. And the y-axis is the one that was inferred with the ICE by counting or by measuring ICE times. And you see the correlation is overall high with the Spearman correlation shown in the black number. However, there are clearly two groups of progenitor states. I should say that each dot corresponds to one progenitor state in our panel of 3,000, each with many, many progenitor states. And the algorithm performs a lot better for progenitor states that are shown in blue and we call well-sampled according to this color key, meaning that if this progenitor population had, let's say, 100 cells, this progenitor state had 100 cells in simulation, how many of those cells ended up having representatives in the sequence cells? 25% means that 25 out of the 100 cells that comprise this progenitor population ended up among their descendants, ended up among our sequence cells. And the progenitor states for whom not many, a very small fraction of their cells had descendants in the sequence cells, the estimates start performing much worse, as you can see here. And to understand why the sampling matters, I have shown you this example. Again, this is the phylogenetic, this is the topology of the example tree we have been using. But let's say this was a progenitor node that actually was an IP2. So it was capable of T1, 2, 3, and 4, as well as 5, which is missing here. But if I do not sample, if I do not sequence all of the cells it gives rise to here, and by chance, because I'm sequencing thousands out of millions or billions of cells, just capture the T1 and T2, I will think this was an IP7, a less potent progenitor state. So when the sampling is low, we end up misassigning progenitor states, and the commitment times that we estimate end up suffering. So now that we have the commitment times and the topology, we combined, again, Eric, another level of recursive combining of information. We combined the information about topology with the commitment time to try to estimate the population size and commitment bias. And this is a little tricky, and I'm going to try to get into it with a little more detail. But basically, on the right, I am showing the topology of the fate map that has been inferred. And on the left, I am showing the phylogenetic, the time-scaled phylogeny. This horizontal line shows the commitment time for inferred progenitor state 5, which we just obtained from the last step. This line crosses a bunch of branches in the phylogenetic tree. And if those branches are across this, their inferred progenitor state of their those branches nodes relates to IP5, those branches basically represent cells, IP5 cells that existed at its commitment time. Let me zoom in to try to explain this better. I have zoomed in on this box in the phylogenetic tree and colored both the nodes and the branches according to this color key on the topology of the fate map itself. So these orange dots were inferred to be IP5. The blue, the yellow ones were inferred to be IP6. But because we know this is IP5's commitment time, and technically IP6 can only exist after IP5, if we are assigning this as IP6, this may be easily a sampling, a stochasticity, or error, right? So we consider all of these branches here that are crossing this and relate to any of these states on this side of the downstream of IP5 to represent cells from IP5. Now we can count on the top 1, 2, 3, 4, 5, 6, 7, 8, and 9 right here before commitment that existed. 36 of those exist across the entire phylogenetic tree. 9 were in this panel. And we call that the population of IP5 at its time of commitment. When we look at where those nodes, where the branches lead them to, we see from the topology, we see IP5 can give you either IP6 or T16, right? We see some of these branches ending in IP6 and the others ending in T16. And we can count them. For instance, 1, 2, 3, 4 are ending in in T16, and 1, 2, 3, 4, 5, 6, 7 are ending in IP6. So this represents the commitment bias, as in 26 to 20, that was the preference of IP5 to become IP6 versus T16. And obviously, we get these numbers across the entire phylogenetic tree. And keep in mind that there are some branches that are not informative due to sampling, and other due to sampling or phylogenetic reconstruction error, which we will get into later. So we next evaluated the performance of this algorithm first for estimating population size. Here you're seeing the log2 of the true population size as simulated for all of the progenitor states in our panel of 3,000 fate maps. And here you're seeing the estimated size as measured by the algorithm I just presented to you in the previous slide. And a great correlation emerges, but even more so than commitment times, there is a separation between progenitor states that are not well sampled among sequence cells and the progenitor cells that are. The correlation is very high for progenitor states that are well sampled, not as much for progenitor states that are not. But another difference that emerges here, fixed sampling is almost uninformative for undersampled progenitor states, whereas proportional sampling, where cells are sequenced according to their abundance, is informative. And this is due to the natural information that proportional sampling captures about the abundance of progenitor states. If a progenitor state is abundant, its descendants are more abundant, and thus the number of those descendants correlates with the size of the progenitor states, typically leading to this correlation that is captured here. The performance for commitment bias is very similar. Basically, you see that for well-sampled populations or progenitor states, there's a high correlation between for both fixed and proportional sampling. Whereas for undersampled progenitor populations, fixed sampling is completely uninformative or near completely uninformative. But proportional sampling is still somewhat informative. And again, because it naturally captures these biases naturally dictate population sizes, which are eventually captured. Eric, are there any questions in this part? Because it's a little tricky.
Yeah, no, I mean, it makes sense. Especially the difference between fixed and proportional sampling does seem like that would make a big difference in your ability to reconstruct population sizes.
Excellent. All right. Thank you. So just to summarize this part of what I have presented so far, this is what we call the ICE-FASE algorithm, which basically uses nodes in the internal nodes of the phylogenetic tree, or a time-scaled phylogeny of sampled cells that have fate informations as chronometers of how these progenitor states are behaving. Then it infers their topology, their hierarchy, and it infers parameters that are commitment time, population size, and commitment bias for each of those progenitor states. And critically, if a progenitor state is not well sampled, let's say its population is very large and not enough cells were sequenced, these parameters tend to lose their reliability, whereas if a progenitor state is well sampled, the parameters tend to be far more robust. Now, everything I have told you so far assumes that we already have a time-scaled phylogeny. And in this case, we had it because we simulated it. It was a perfectly accurate time-scale phylogeny. But in real-world experiments, we are going to have to get this from barcoding data. And this is actually very important because so far the performance you've seen of the ICE-FASE algorithm is for perfectly accurate topology and tree topology. And those of you who are familiar in the field, familiar with the field, you know that error in tree topology, reconstruction, and inference is very uncertain, and we basically can't predict how error affects the results of an algorithm. So it's important to assess whether ICE-FASE algorithm can actually perform on topologies that are less than accurate. But for reconstructing topologies based on barcodes, it's important to keep in mind a few important principles. And those of you with phylogenetics, evolutionary phylogenetics background may find this very interesting. There are some key aspects between cellular phylogenetic mutations and evolutionary mutations that, in our view, warrant having a unique and having a system for phylogenetic inference that best suits the system. One is that the initial states are known for every site, as in we know in the zygote or whatever other developmental system, what is the non-mutated allele. So the initial states are known, or the earliest ancestral state is known. Mutations are reversible, irreversible, which is very important. Once a site mutates, you can basically assume it doesn't mutate further. Whereas in evolutionary phylogenetics, a site can mutate, let's say from G to A, and then in one sublineage back from A to G, which makes things very complicated. Mutation sites are independent, and they are not under selection in lineage barcoding. This is critically important and very different from evolutionary phylogenetics, where they are under selection and they're not necessarily independent. You often see different mutations and variations in different regions being correlated, whereas here in this system they are not, because they are not functional. So keeping these in mind, we wanted to establish ways to infer phylogenies from lineage barcodes. However, to do that, we needed to also simulate a number of lineage barcoding outcomes in our panel of fate maps. And what I'm showing you here is results from actual experimental data in mice that shows the behavior of these barcoding sites over development. You see time here. Each line corresponds to one of about 60 different barcoding sites that were analyzed in a mouse model we often use, and the y-axis is the fraction mutated. And they generally have a constant rate of mutation that accrues over time. So based on this, we established a barcoding model where a barcoding site mutates at a constant rate according to a Poisson point process. And once it mutates, it gives rise to a number of alleles, a number of different mutant alleles, and each with its own emergence probability. So in this model, if you have a phylogenetic tree, you can basically start the barcoding site as unmutated here and simulate as it goes down the phylogenetic tree. And once a mutation happens, for instance here, everything downstream gets that mutation. This is just one barcoding site. And we estimated these various parameters actually from the data, and we validated that our parameters basically recapture what the data has. So now to come back to this panel, we already had simulated phylogenies, 3,310 phylogenies for this panel of fate maps. Now, on each of those phylogenies, we simulated barcoding using the model I told you, with 50 barcoding sites in every cell. 50 in every panel, those 50 would be identical. And this is an example of the phylogeny. This is actually the phylogeny I showed you earlier. It's just sideways. And this is the barcoding outcome that was simulated with our model based on this phylogeny. But the reality is that this is what we would get in a barcoding experiment, and we would have to infer the phylogeny. So to To infer phylogenies, we took advantage of the unique aspects of cellular phylogenetics and lineage barcoding that I told you about earlier. When you consider two cells with their observed mutant alleles for a particular barcoding site, the probability of observing those alleles can be expressed as a sum of two terms. Either the mutation happened before their most recent common ancestor, in which case they both have to have the same allele, or the mutation happened after their most recent common ancestor, in which case the probability can be calculated based on what mutations have been observed. Now, the beauty of this is that these two cells obviously don't have just one barcoding site and its respective mutations. In our case, they have 50 barcoding sites and their respective mutations. But because those barcoding sites are independent, their individual probabilities as a function of when this time since most recent common ancestor is, can be obtained by just multiplying the individual probabilities for each site. So as a function of how many barcoding sites there are, what is each of their Poisson point process mutation rate, and what is each of their allele emergence probabilities, as well as the alleles that are observed in these two cells, and the time to the most recent common ancestor, we can get the probability of observing the entire allele sets between two cells. Now, if we maximize this probability or this likelihood as a function of the time to most recent common ancestor, we basically get a measure of temporal distance between each pair of cells, or we get a measure of what is the most likely time that separates these two cells from their most recent common ancestor. And then we can apply UPGMA style clustering algorithm to those pairwise times to reconstruct a tree. This is an algorithm we call phylogeny inference using likelihood of time, or Phylotime, and I will try to make it more clear with a few plots. So this is the barcoding outcome here. As you have seen before, every row is a cell, every column is a barcoding site, and the colors are the mutations, and the cell types are known. This is the matrix of pairwise time to most recent common ancestor between all of those cells, estimated using the two-cell model probability time to most recent common ancestor probability model that I presented in the last slide. And this is the inferred phylogeny that is done by that is obtained by UPGMA clustering of this matrix. Now let's look at how this Phylotime algorithm performs. These plots show the performance of the Phylotime algorithm. Let's first focus on the left side, where we are just comparing only the topology of the tree that has been inferred using Phylotime to the topology of the actual simulated phylogeny, the underlying true phylogeny. And simulations have been done with 25, 50, or 100 barcoding sites. And to compare topologies, we use Kendall-Collian distance with a tuning parameter set to 0, which basically compares topologies. In red, you see Phylotime's performance. In green, you see a Hamming distance based method performance that just counts the number of mutation differences between two sites. And in blue, you see performance of Cassiopeia, a maximum parsimony algorithm. I should add that this is Cassiopeia 1.0, and it wasn't exactly designed for this scenario. And Sebastian, who will be presenting in a couple of weeks, I suspect will present the most recent versions of Cassiopeia. But we couldn't really compare Phylotime to too many things, because here we have 530 trees, each with 1,600 terminal branches. These are from the 53 fate maps with 16 terminal cell types. And there are not many algorithms that can actually handle this. Phylotime can, the Hamming distance can, and Cassiopeia can. So that is the comparison that is taking place. And you see that based on topology alone, Phylotime predicts reasonable topologies, and it converges when there are more barcoding sites, which is reasonable. But when you look at the combination of topology and branch length, which is shown here on the right, this is done by using the Kendall-Collian distance with a tuning parameter set to 1, which consider this distance is 0 if two rooted trees have not only identical topologies, but identical branch length. And you see that only Phylotime captures fairly accurate tree topologies plus branch length. And importantly, it converges to 0 error when there are additional barcoding sites. So we concluded that Phylotime, which also happens because it's clustering, it performs in polynomial time. It performs in polynomial time, and it can handle a large number of barcoding sites, is a reasonable algorithm to apply. Now, the question becomes, given the uncertainty in reconstruction of phylogeny and the fact that even Phylotime reconstructed phylogenies are not perfectly accurate, they are not exact matches for the true phylogeny, does the ICE-FASE algorithm still work, or is it so sensitive to any error that it completely falls apart? And this result shows the application of Phylotime to our panel of 3,310 simulated phylogenies, followed by ICE-FASE algorithm applied to the inferred phylogeny and compared with the true phylogeny. So this on the left, you see progenitor states with sampling fraction that was low. On the right, you see all the for all the progenitor states that with adequate sampling fraction, and then this row is for fixed sampling. This is for proportional sampling. Dark blue is Phylotime phylogeny, and light blue is true phylogeny. Basically, what you're doing here is really just comparing the dark blue, which is Phylotime fate map reconstruction using Phylotime, to true phylogeny, to fate map reconstruction using true phylogeny, and seeing what fraction of correlation was recovered. In general, Phylotime is recovering more than 90% of the correlation with true phylogeny. And this result shows that ICE-FASE is stable and does not require perfectly accurate phylogeny, which is nearly impossible to obtain. And moreover, that the phylogeny that Phylotime infers is accurate enough for these purposes. So to summarize in one slide, all of what I have told you so far is that we have established a Phylotime algorithm which takes the lineage barcoding outcomes from a number of independent barcoding sites, which could be synthetic barcoding or natural somatic mutation-based barcoding, and obtains a time-scaled phylogeny from them. Then the ICE-FASE algorithm uses this time-scaled phylogeny and the nodes within it that are important for that have information about fate decisions to reconstruct the hierarchy of the progenitor states that led to these observed cells, observed terminal cells, as well as decipher some parameters about important parameters about each progenitor state's behavior, which are its population size, commitment time, and commitment bias. Almost everything I have told you so far today has been published on bioRxiv in this paper, and all the relevant data sets and codes are available on this GitHub repository. I want to close by acknowledging my lab members and our collaborators who have contributed to this project. People whose name with has an asterisk here contributed to this project. Obviously, Wei Xiang led the whole effort. Abel contributed to the Phylotime algorithm development. Kathleen Kathleen and Claire contributed to aspects that I didn't discuss much. Likewise, Soichiro. Honkai was the co-advisor on the project. With that, I thank you, and I'm happy to take any questions.
Thank you, and thanks for persisting through the cold. As you know, I a little under the weather as well. So I hope to get some comments and questions on YouTube Live. I guess just a general question: what are some of the things of the aspects of this process that don't feel captured by your simulator?
don't feel captured.
Or don't that aren't captured well by your simulator.
Sorry, is it are or are not?
Are not.
Are not captured well by the simulator. Well, I think the simulator captures what we put in it, and we haven't put everything in it. For instance, one of the things we haven't captured very well yet is so far when we say a terminal type that you have isolated, let's say T5, we are basically assuming this is a cell type of a pure progenitor state history. But what if another route through the progenitor state gave rise to the exact same cell type in a way that we couldn't separate? Now, you could argue with modern molecular approaches, this is increasingly rare, but that's an aspect that I don't think we capture very well and would be very interesting to evaluate.
That was going to be another question. So it does happen that you have different lineages converging to the same cell type.
Yes, it does happen that different lineages convert into the same cell type. Very strict epigeneticists might tell you that even if from what you can tell, those cell types look identical, they may not be identical epigenetically because they went through different routes in the epigenetic landscape, or what some people may better know as the Waddington landscape, and there likely remains some vestiges of that different route in each of them. But for practical purposes, it's perfectly possible.
I mean, I guess one thing I was thinking about, and we played with this type of data several years ago, is it seemed like, at least in the system that we were looking at, it seemed like there was some non-independence between the barcodes.
Yes.
And simulator seems to...
and so I guess I said Philotime's assumption is the barcoding sites are independent. And it's great that Sebastian is giving a talk in 2 weeks, because the type of synthetic barcoding that we have implemented in my lab has implemented in the mouse, the barcoding sites are actually independent. But some of other very interesting and effective barcoding strategies do have codependent barcoding site. Basically, the barcode is an array, a physically contiguous array of barcoding sites where a mutation in one can cause a mutation in the other. In such a case, Phylotime wouldn't be applicable, at least as is. But I know your group and other groups are developing models of understanding how these contiguous barcodes work and how to maximize the information derived from them. But it should be emphasized that not all lineage barcoding approaches involve independent sites.
Yeah, I mean, I guess, though, even if they are sort of, I'm guessing these are like spatially separated and things like that, could there be a sort of a latent state that is like determine something about how much the barcodes are going to get trimmed, even if they're not contiguous? Could there be something like some factor gets upregulated and then it makes it more likely to have two trims, even if they're separate?
That's a great question. So I'll break this down. I think this question needs to be answered for each barcoding system separately, right? One barcoding system that is universal is somatic mutations, the most expensive, hardest one, but the one that is most universal, which are naturally accumulate. And for those, you can generally say they are independent. They are just the probability of a polymerase to make an error or a DNA, a certain kind of DNA damage based on oxidation, for instance, to happen. And that probability is, as a first order approximation, constant over time. Right. For our barcoding system, which I didn't talk about much here, but there are these things we call homing guide RNAs. They are very, very small synthetic circuits. They express one tiny RNA using a constitutive promoter. It's actually a promoter that drives tRNA expression normally, which is not subject to much regulation as far as we can tell. And then in presence of Cas9, this thing comes in and basically makes a mutation in its own position. And we have tens of these scattered in the genome in different locations. As far as we can tell, they operate largely independently. Based on the data we have, they also operate at the same rate independent of cell type. The reason for that is that we don't think the promoter that drives these small guide RNAs is regulated highly. However, Cas9 may be regulated in different cell types. We know that the transgenic mice that have Cas9, different levels of Cas9 may be regulated, expressed in different cell types. But as far as I have seen, and as far as we have been able to find in early stages of embryogenesis, at least the first half and then some of embryogenesis, Cas9 tends to be uniform. And the differences that are seen is mostly in the adult tissues. So one should consider those caveats that you're pointing to, depending on exactly which system they're analyzing and when that system developed. Could Cas9 or homing guide RNAs have been regulated differentially in those systems?
Yeah, I mean, and it feels like maybe you like because you have sort of a constant time Poisson process assumption for your timing, and so that seems relevant.
Absolutely.
So I just have to ask, so you're saying that basically methods that are based on Cayman-Sokal parsimony are not scalable to 1600 tips?
No, I don't think I am saying that. And I think even, well, it depends. If you have to do a tree search, it becomes very difficult. And there was a recent paper that they also used sort of cloud computing to or distributed computing, I guess, to search very large tree spaces. But even Cayman-Sokal parsimony has a mode that assumes that the ancestral state is known, I believe, and I suspect in that mode it would be scalable. I don't think in that mode it would give you a sense of time, though, necessarily, as in the branch length you would get.
No, that's true.
And
yeah,
but I mean, everything. So I think the sort of what we believe is sort of the magic of time is that, let's say you have sequenced 1,000 cells in an organism, right? With this quantitative fate mapping, that I'm showing you, what it is that we think makes it so powerful is that you have 1,000 cells sequenced. You get 1,000 choose 2 estimates of time, right? Each one of those estimates may not be perfect. In fact, it isn't. But when you put that, all of them together, they embody a lot of information that sort of in resilient ways captures the emergent features of the progenitor landscape that gave rise to those 1,000 cells. Right, and that is what I'm not sure if, let's say, that type of parsimony can capture.
Yeah. I mean, it's just it might be a little bit more resilient to some of the, like, if there's some sort of problem, like systematic problem with time estimation for parts of the tree, it might help with that. But who knows? But we have a couple questions. Thank you. Sebastian Priot asks, thanks, Reza, for the awesome talk. One question, one tricky aspect about lineage tracing data is missing data. How does the tree reconstruction method you presented deal with missing data?
Fantastic question. This actually, to those in the field, this is one of the first questions that comes up every time, because all real data have missing data. So what I'm showing you here on the left, for those of you in the audience who may not be familiar, you see that there's a color in every pixel. And when you get real data, that actually is not at all what it looks like. And there tends to be, depending on the method, anywhere from 10% and sometimes up to 70% of these that are missing. They're just gray. And we have assessed this, and hopefully we will publish that soon. But Sebastian's question is, how did we deal with it? Weixiang actually established a very interesting machine learning strategy based on XGBoost that sequentially and iteratively imputes the missing data from existing data. It goes from the sites with the least missing alleles to the site with most missing alleles, and then trains a machine learning model on all the position on all the cells that do have an allele for that site, and uses that to impute for the cells that don't, and then goes from the least missing to the most missing. From there, I think that GitHub repo has that actually. And a natural follow-up to that question would be, what happens if you have in barcodes missing data? By and large, we see, obviously, if you have missing data, the more missing data, the higher the error of any estimate that you obtain. But the ICE-FASE and Phylotime algorithms, both of them behave stably in that the error increases gradually and not drastically. In general, it seems to us that if you have 30% missing data, it does depend on a lot of things, including the number of cell sequence and as well as the number of barcoding sites that you have. But if you have 30% missing data, up to 30%, you can make fairly robust inferences. And if you have a lot more than that, there's not, you can't. But this depends on a lot of things. I perhaps shouldn't even have said any value, because it depends on many things.
Cool, thanks. Anna Zhukova asks or says, thanks for the great talk. In classical phylogenetics, neighbor joining gives better results than UPGMA for distance-based tree reconstruction. Could it be used in cellular phylogenetics?
I think it would be used. I think it can be used. I don't feel like I'm the appropriate person to answer this question.
Shouldn't be hard to, I mean, there's definitely scalable neighbor joining things. It would be worth a try. I mean, I could say that definitely the situations in which neighbor joining does much better than UPGMA is when you have very non-ultrametric trees. And so I guess.
And these tend to be almost by definition ultrametric trees.
Maybe it's worth a try. It's a great suggestion.
Yeah. One thing I would say is that if Wei-Xiong was answering this question, at least one of the things he would say is that he tried multiple things to see what performs best. Neighbor joining may have been among them. He may have also had other reasons for UPGMA, but that's a great question. And Eric provides a great answer, but I can't add to that.
I'm sorry, I feel like we should end, but I just have to really ask one final question, which is, this feels so much like gene tree species tree estimation. Are you familiar with that line of work? Gene tree. So trying to estimate species trees from gene trees. So basically, when you have
different barcoding sites are different genes that provide you with different trees, and what you want to basically obtain a tree that explains the entirety of the behavior.
Exactly. And you have this thing that, so like the modern gene tree species tree estimation techniques allow this thing where you have you can have multiple lineages that go up and they're the same ancestral species. So, and that corresponds exactly to your situation where you have multiple cells dividing in a progenitor lineage that haven't fully differentiated into their respective lineages. They also provide natural estimates of population sizes and sort of everything. It feels just like, I mean, I'm probably missing something, but it feels like this is very much exactly what these methods do. I mean, the limitation, and I think that these things are usually implemented in a Bayesian context, and the Bayesian methods of course have limitations as far as scaling is concerned. So they yeah, I don't know.
Is it that you think a Bayesian framework for estimating a timescale phylogeny based on the gene tree style methods may also be applicable here?
I think it could be, but it's sort of like, I guess here, even you could, at very least, it's worth looking into. I mean, I guess maybe you only have one tree, but you're trying to fit it into one species tree, because at the end of the day, you have one tree of cells.
Yes, you only have one tree of cells, and you have irreversible mutations.
Okay. Yeah, maybe
But
that's a bad idea.
I think it was still very interesting to try.
Well, anyway, I think there's some older phyloseminars on the gene tree species regression. There's certainly some good review papers out there. But anyway, I never miss an opportunity to make a fool of myself on YouTube. So there we go. Thank you so much, and I really appreciate you giving us sort of a bit of background so that people could like sort out what's happening with future talks. So
Thank you
thanks
very
again.
much for having me, and I'm very excited about the upcoming future talks.
Yeah, me too. Thanks.
Take care.